Alright, here we are talking about phylogeny and speciation. And this has to do with the classification of species and how different species became their own separate species from common ancestors. So let's just sort of get right into it. This is going to be a little bit of a long one, but that's all. Phylogeny has to do with the evolutionary history of an organism. So basically, you're looking at where an organism is now and where it came from, and all the different relationships that occur within that organism and other species. And evolutionary scientists look at this as a really important part of the science because it sort of gives you a timeline as to where, why, and how things came about. And when these things are organized, they're organized into something called a phylogenetic tree. And we can create phylogenetic trees in many ways. A lot of times they're usually done by the structures of an organism, maybe how it behaves, but even more accurate would be biochemical attributes, where you're actually looking at DNA and proteins for the organism. So when we create a phylogenetic tree, the lineage is the series of ancestors. And this is kind of like a family tree. It's just sort of a picture of where things are. Anytime we see the lineage split, we call that a node. And nodes are going to lead to more and more branches, trees that show different relationships between different organisms. Phylogenetic trees can get very big and very complex, but the one thing that we need to know about these is that depending on who created the phylogenetic tree, we could put all the same organisms into the same pool and get multiple different trees based on who's looking at them. When we talk about the very oldest person, the most ancient ancestor, we call that part the root of the tree, and we'll look at and see some of these examples right now. So when we are looking at this, let me pull out the pointer, we got our root right down here, and if you notice down on the x-axis over here, we have kind of a timeline. Anytime that we see a node right there, there, and there, that corresponds to a different time. And at the node, that tells us there was a common ancestor between two organisms that split off from there. So chimpanzee and the human had a common ancestor about 5 million years ago. That common ancestor had one with the gorilla about 7 million years ago. And with the orangutan, we had a common ancestor somewhere around 13 million years ago. So this gives us sort of a time component and allows us to see when different lineages sort of split off and what these specific speciation events might have been. What's the way that new species are made. Just some basic terminology. We have taxon is the end line, and that is basically an individual species is a taxon. Plural, that would be taxa. So if we're talking about multiple different end lines, we're talking about taxa. A clade is a group. So when we look at the phylogenetic tree, it's a group that has common ancestors. So the entire phylogenetic tree could be a clade, or we could look at smaller individual clades on the large phylogenetic tree. Your sister species and sister clades are just either the individual taxon that's your closest relative or the group of taxa that are your closest relatives. So why do we use phylogeny? Well, kind of the basic stuff. We want to organize things, and we want to see how things have changed and possibly use it for predictions. That's what the flu shots, they use phylogenetic trees to figure out where the virus is going to change, and they use molecular clocks and all the stuff that we're going to be talking about in a little bit. When we are comparing organisms, any trait that was present in the ancestor or the root is something that we call an ancestral trait. So anything that's still around that came from a long time ago is what we'd call an ancestral trait. Anything that's different from something that the root had, we would call that a derived trait. And when we talk about ancestral and derived traits, some things have similar evolutionary origins, some things don't have similar evolutionary origins. Anything that has a similar origin, we call it a homologous structure or homologous trait. So an example of that is going to be when we look at these bones of the bird and the bat. Now, there's a green sternum there and a green sternum there. Clavicle scapula in the bat, clavicle scapula in the bird. Two humerus, radius ulna, and then kind of hand things. The bones in this picture are actually homologous. The actual wings themselves, however, are not homologous. They are due to something else. So that's where it gets slightly confusing at times. But these bones had a common evolutionary origin. The colored ones, they came out at the same time in a common ancestor. And anytime we have a derived trait that is shared by more than one taxon, we call that a synapomorphy. Synapomorphy literally means a shared derived trait. And here's a couple examples of synapomorphies that we see in organisms today. Now, like I was saying with the wings of the bats and the birds, they did not evolve together. So they evolved on two completely separate planes, but that's because they have similar living environments. They have similar stresses acting on them. Anytime you have different evolutionary origin but similar structure and function, we're going to call that convergent evolution. So anytime you have two unrelated things that evolve the same structure, that's convergent evolution. I always think of dolphins and sharks. They look very similar because they live in the ocean and they're carnivores and they do similar things. They swim, they eat. But they evolve those body shapes at very different times because dolphins are mammals and sharks are fish. An evolutionary reversal happens when a derived trait isn't successful for one reason or another, and the organism actually ends up back in an ancestral state. We talk about whales having hip bones. Well, a whale swims all day, which leads scientists to believe that at some point whales probably lived on land, or a distant ancestor of whale lived on land, and eventually it moved back towards the water. So that would be an evolutionary reversal. And a homoplasy is just a fancy word for any trait that's generated by convergent evolution. So a wing and a bat and a bird would be a homoplasy because it's brought about by convergent evolution. When you are building a phylogenetic tree, we have to make some assumptions. We want phylogenetic trees to look very simple. We shouldn't be really confused as we're looking at these things. So usually when we make a phylogenetic tree, we always have what's called an outgroup. An outgroup is a species that was related to the root, but very different than everything else. And it's usually just sort of on a line by itself, and we'll see an example of that in a second. We also want to assume that derived traits came about only one time because that just makes our life a lot simpler, and we'll talk about that simple principle in a second. When building a phylogenetic tree, a lot of times we'll use a chart that looks something like this, where we have minuses and pluses. On the left side of this chart, we have all of our different species, and across the top we have different derived traits. Well, if it has a minus, there's no derived trait. And look at this, the lamprey, which is our outgroup, which is one that, if you go back to Sesame Street, which one of these does not belong? Well, that's your lamprey right there. It's got none of these derived traits. Everything else has a few plus signs down here. And when we're talking about evolutionary origins, the more plus signs you have in common, the more closely related you are. And that's sort of how we create the phylogenetic trees, is start with a chart that looks something like this, and then we put that onto paper. And when I look at this, I have my root or my common ancestor right about there. And then each one of these derived traits gets put here. So the jaw. The lamprey doesn't have a jaw, but everything that came from this one lineage is going to have a jaw. Now, if I go left, I get to a perch, which has a jaw, but the perch does not have lungs because perch use gills. So everything from this point down on the lineage has lungs, and so forth. And we get going down here. So these two would be sister species. The crocodile and the pigeon would be sister species. If I were looking at a clade, I would include maybe a common ancestor with these lizard, crocodile, pigeon would be one clade, and their sister clade would be the mouse and the chimpanzee. So if we're using some of those terms that we learned a little bit ago, that's sort of how they would work on a picture that looks like this. And these are kind of our basic principles when we're looking at creating a phylogenetic tree. We use what's called principles of parsimony, or the parsimony principle. That basically the easiest way you can explain something is the best way to do it. Remember, depending on who's making the phylogenetic tree, it could look completely different. If I give two different people the same groups of organisms, it could be completely different. As long as they can simply explain it, then they're doing what they're supposed to do. And Occam's razor just is basically, if you look at something, you should know what it means. You shouldn't have to guess, and that's basically what these two things mean. Keep it simple, keep guessing out of it. So, what are some sources for our phylogenetic trees? Well, morphology is the shape of things, size and shape of things. And that's kind of the basic starting point that we usually use, is what does something look like? If it looks the same, let's go down to it. We look at developmental stages, seeing if things age the same way. I give the example of a butterfly and a moth both start as little caterpillars. It makes a cocoon, and then the winged organism comes out. So it would kind of tell us that they have similar evolutionary origins. Do they act the same? And kind of the big one with biology now is the molecular data. Now that we can sequence DNA and look at the actual structure of proteins, that's probably going to give us the best idea of how closely related things are, because that gives it down to, I mean, we're looking at genetics, which that's the basis of everything that we do in evolution is genetics. Now, what else can we use? Well, maximum likelihood. Maximum likelihood is a mathematical method to see how close we are and how often these molecular changes are happening. And we can use maximum likelihood to make predictions on where a tree might go in the future and try to figure out where something came from in the past. Um, I'm not going to ask you guys to actually figure out the math behind this. That's a little bit above our league. But just understand, maximum likelihood has to deal with math and figuring out where things came from and where they're going. We can use phylogeny to reconstruct ancestral traits, and everyone wants to make mammoths come back for some reason. I don't get it. They had their chance, in my opinion, but hey, mammoths, we find really good preserved samples of them in ice a lot of times, which gets people a little excited that maybe we could bring back a mammoth potentially. But understanding the phylogeny of the mammoth and the family and the organisms that came after it, we can sort of reconstruct what mammoths looked like and what they did and how they. Um, we can use it to date things. The book gives you this example of following HIV and the different changes that the virus that took place in the virus in different years that those happened, and we can see all the little nodes and when they were going on and how close the DNA is to the ancestral. Well, when we put that in with something called the molecular clock hypothesis and we start plotting these things out, we get a line of best fit. And this smaller graph on the left actually belongs right here. It's just kind of an out, just kind of took a section out of that. And this line of best fit, can be added on to this original graph, and we can see that HIV started probably somewhere in this time range. And that's using molecular clock, maximum likelihood, and an understanding of how phylogeny works to sort of trace back the roots of certain things. In more recently, we looked at the Ebola virus, which there was a big outbreak of this in 2014, 2015. And following that back, we can use molecular clock hypothesis to figure out where Ebola started and how that got going. So one of the biggest things for us with phylogeny is classifying things. And Carolus Linnaeus is a scientist. He was a botanist, actually, and he wanted to classify flowers, and he wanted a better system. Because before that, basically people looked at organisms and said, well, we'll classify you based on where you live. So everything that lived in the water was related. Everything that lived on land was related. Everything that flies was related. Well, we talked about birds and bats. Birds are birds. Bats are mammals. Two very different families. Sharks and dolphins. You have fish versus mammals. I'm not very closely related to a snake, but we both live on the land. So Carolus Linnaeus wanted to get a better system, and he came up with what's called binomial nomenclature. Binomial nomenclature basically means that you take you create scientific names for organisms and you base it on classification. And in order to do that, you use a taxonomic tree. This domain up here is the most general category, and there's three domains. You got the eubacteria, you got or prokaryota, you got the eukaryota, and you got the archaea. And then from there, all the eukaryota, which is what we fall in, has different kingdoms. You could be Animalia, Plantae, Protista, or fungi. Fungi. Ah, I'm a fun guy, so, well, I'm not a fun guy, I'm an animalia, but that's okay. And then we just sort of get things going a little bit more until we get down to the specific epithet, which tells us where our individual species is. When you're writing binomial nomenclature, we only write the genus and the specific epithet. The genus is always capitalized. You can abbreviate the genus if you want. Sometimes you can put the scientist's name at the end of it, so you could put like Homo sapiens Linnaeus or H. sapiens Linnaeus because he's the one that named it, and obviously it goes after the scientist that named the group. But that's a really easy way to classify things and sort of give things a hierarchy as we are looking at them to help organize things and make it easier for scientists as they're studying these organisms. When we are looking at classification and we look at a phylogenetic tree, we can look at different groups on a phylogenetic tree: a monophyletic group, a polyphyletic group, and a paraphyletic group. Well, I'll be the first one to tell you that these bottom two are considered inappropriate because they don't look at all the evolutionary relationships. Monophyletic, you have common ancestor and every descendant from it, and we'll see an example of this on a phylogenetic tree in a second. Polyphyletic, no common ancestor in the group that you're looking at, and a paraphyletic has a common ancestor, but not all the descendants. So let's see what that looks like on an actual phylogenetic. If we look here, we have a common ancestor right there, but we have 3 organisms out of the 4 that came from that common ancestor group. So that's going to be a paraphyletic group. This one, there is no common ancestor highlighted between these 3 organisms, so that's going to be a polyphyletic group. And the bottom one has 1 common ancestor and all the descendants, so that's going to be our monophyletic group. And that's sort of what we want to focus on if we are scientists studying a tree. If we're looking at a group, we want to look at the common ancestor and everything that sort of came from that. So we've been talking about different species and all these splits and nodes and sort of how it's sort of organized in phylogeny. Well, that should lead you to the question, how do we get a new species? How does this all happen? Well, that's a great question, and the way that that happens is through a process called speciation. Speciation is based on the idea of reproductive isolation. Reproductive isolation means 2 organisms can no longer exchange genes, meaning they can't breed and make viable offspring. They may be able to make an offspring, but if it can't have a baby, basically their genome dies with that offspring. So if you can't produce viable offspring, it's said that you're reproductively isolated. And once 2 species become reproductively isolated, we say that they are 2 separate species. In order to look at what a species actually is, we have a couple species concepts. Linnaeus used a morphological species concept, which is okay. It basically looks at shape and structure. If I look at these 2 groups of species, well, these 3 ducks up here, male, male, female, they're all the same species. These are what we call dimorphic species because there's 2 shapes or 2 structures or 2 color patterns for the same species. These 2 frogs are what we call cryptic species. They're called cryptic because they look very similar, but they are not related at all. They can't actually interbreed. So we got a little bit of a problem there. So these ones can pose a little bit of a problem, but using morphological species concept is a good starting point for the most part. What we need to do is go a little bit deeper, and there's also the biological species concept, which has to do with the potential to interbreed. Basically, Ernst Mayr was the scientist that sort of came up with this. He said if you can't breed with something else, you're not the same species, which is sort of where we are today. So this was kind of a acceptable addition to what Linnaeus was doing. And if we want to get a little more complex, keep going, then we can use what's called the lineage species concepts, which has to do with puts a time component to this. So realistically, when we look at our 3 species concepts, we kind of use all of them, to be completely honest, because we look at sizes and shapes and color patterns and all that, then we try to figure out if they can breed, and then when we put them on the phylogenetic tree, we put in that time component. So kind of all 3 of these get used by scientists today. So let's get back into the splitting. Well, this is a brief review of, I think it was chapter 7 or 8, when we talked about cell division. It was meiosis, where we start with a diploid cell to begin with, and we end up with 4 unique haploids. And these haploids have a unique combination of DNA, which makes them special because each haploid gamete will fertilize itself another haploid gamete, and that'll create a diploid zygote, which will eventually turn into an organism. So Dobzhansky and Muller came up with an idea of how this part actually happens. And basically what they looked at was you have a species. For some reason, this species gets separated into 2 groups, and we'll talk about reasons why they separate into 2 groups. There are stresses that happen on this species, there are stresses that happen to this species. or this group, I'm sorry, this group and this group. Over time, changes will happen in the DNA that changes will happen in this top group that never happened to the bottom group. Changes will happen to the bottom group that never happened to the top group. And eventually the DNA will change enough where, as we move forward, these two can't get back together and they can't create viable offspring. And once that happens, then we say that that group has become speciated. They become reproductively isolated and they create two new species. Centric fusion has to do with an actual reshaping of the chromosomes. And again, one group initially, the group splits off into two subgroups. This subgroup has some changes in DNA shape and structure. This group also has changes in DNA shape and structure. And eventually, those get bred into each of those groups and they can't interbreed. So they become reproductively isolated through centric fusion, which is a change in how well something can interbreed with its original part. Well, depending on the species and depending how large a species is and how isolated they get, well, this can happen really quick. This might take a long time. It completely depends. Um, some examples of things that can separate those two groups, one of the most common is allopatric speciation. Allopatric speciation happens when there is a physical barrier put between something. Let's say you live on an island and there's a lava flow and you can't get to the other side of the lava, and half your species is on one side of the island, half is on the other side. Well, eventually nature will take a course. They'll start breeding with each other. They're going to wait till the lava goes away, so they start breeding. And if changes happen to each of the groups and they get back together and they can't interbreed, then we say that they've been speciated. Some other ways this can happen is continental drift, sea rise. This is all stuff we talked about over the history of the world that's happened since the world was formed. Our world's undergone a lot of change. So these speciation events happen throughout the course of time. Sympatric speciation happens when you have a separation without any physical isolation. And there's a couple ways it can happen. One of those ways is when polyploidy happens. And polyploidy happens when chromosomes get duplicated. Well, a couple ways that chromosomes get duplicated. One way is autopolyploidy, which auto means self. Something happens where the duplicated chromosomes the chromosome number gets duplicated inside an individual species. A lot of times this is an error in meiosis, and very common in plants. We talked about this a few chapters back, where plants can self-pollinate. And anytime you can do self-pollination and things like that, it makes it really easy for you to become tetraploid, because if you're a tetraploid organism, because your genome's been doubled, you went from 2N to 4N, you can only successfully reproduce with other tetraploid things. So if you have a tetraploid offspring that can make babies with itself, that's going to really reinforce that autopolyploidy and potentially create a whole new subgroup. When an organism has been reproductively isolated, they might cross back and say, hey, let's see if we can rekindle some old love. Well, anything that can, if the organism gets back together with that other subgroup's members and they can't reproduce, whatever it was that caused that isolation in the first place is reinforced, which means whatever it was that caused that isolation, whether it was some kind of continental drift, whether it was a lava flow, whether it was duplication of chromosomes, that got reinforced because it worked. It did what it was supposed to do. So when we look at things that can prevent hybridization, anything that actually prevents the fertilization of sperm and egg is called a prezygotic isolating mechanism. Prezygotic means anything that happens before you create a zygote, which hopefully we know zygote is sperm and egg fertilizing each other. Well, mechanical isolation basically means the reproductive organs don't work together anymore. The bits and the pieces aren't lining up, they aren't matching, they aren't fitting. Something isn't happening that's preventing the organisms to mate and make a baby. Temporal isolation has to do with seasons and timing. If one group decides they only want to mate in the spring, the other group decides they only want to mate in the fall, well, that they have created two separate groups. That's going to prevent isolation. It means the spring people won't mate with the fall people, and eventually you'll get separate lines. Behavioral isolation has to do with females choosing mates based on certain behaviors. And if something makes a noise or has a presentation that the female doesn't like, they'll stop mating with them. And that is behavioral isolation. They're basically looking at for specific things in their partners. Habitat isolation has to do with just simply moving. Maybe one group wants to move to the trees, one group wants to stay on the ground. One group goes up to the mountains, one group stays by the ocean. Whatever it happens to be, the habitat gets isolated. And gametic isolation means the sperm and the egg don't jive together anymore. They don't work with each other, and if the sperm and the egg don't work together, then you're not going to create a zygote. If a zygote is created, but there's no, the offspring doesn't go for a variety of reasons, that's called a postzygotic mechanism. With low hybrid zygote viability, that basically means the zygote never is born. It doesn't survive long enough to even be born. With low hybrid adult viability, the organism is born, but it never makes it to sexual maturity. With hybrid infertility, it makes it to adulthood. It can have sex, which is a combination of a horse and a donkey. They're sterile, they can't have babies. And all of these things reduce fitness of an offspring. And hopefully remember that fitness means your ability to survive and reproduce. And finally, sometimes once you've made a separate species, sometimes that species may be able to make some offspring. And an area of overlap where two different species live, where they can actually make some viable offspring, even though they're two separate species, we call that a hybrid zone. And this only happens when you have probably organisms that haven't been speciated for very long, but they are still able to make a viable offspring. Well, hopefully you enjoyed that, and if you have any questions, ask me in class. Have a good afternoon, and thanks for listening.